too much since the last time we talked. So I figured we could go through whatever kind of open questions you guys have and see what we can generate in the next.
I think the one question is the files that they combined 4 files put together. I guess it's the 165, 166, and then the gene expression, the transcriptome, and the heavy, the immunoglobulin expression. So, and are they together? Because I can never get that figured out. And right? Can we put all that together and then, because in the CVS file, and then open it up?
CSV, right.
CSV,
After we concatenate and then export to a CSV, then I sort of had some difficulty to figure out which sample is from which cell is from which sample, that kind of question.
Well, I have kind of trouble opening it up, the combined file. Oh, good, you got it. So if they open up in your hands, and especially in the, the BDJ.
Right, and also the BDJ, the CSV file, the proper way to edit it, so it will upload to the BDJ and it will run.
Right. I have a lot of trouble with BDJ. Maybe I don't have the right version. Not a lot of trouble. Just doesn't open.
I didn't copy all of that. I did hear, John, you give a little description of the combined files and their associated metadata.
Yes, right.
The metadata is certainly separate from those expression matrices,
Okay.
but the two combined files, those are the two expression matrices combined.
Which one's that? The BDJ Explorer?
No, so this file, concat 165, 166,
okay.
that's just a combination of the two gene expression matrices, the whole transcriptome.
Oh, okay. What about the BDJ part? Is that
the BDJ component is what is fed into the BDJ Explorer.
Oh, okay. But that's in the combined, because what you want to do is try to analyze the transcriptome and then the BDJ associated with that at sort of the same time, or the other way around. We could analyze BDJ and go back to the transcriptome. You know what I mean? So have it all linked together and then compare, for example, BDJ of 165 to 166, and then go back to their genes that are under a particular difference in BDJ. If we find the VDJs are different by some criteria, and then we go back to what genes are different.
Right. We can use information from the mapping, this mapping between the VDJ information and the gene expression matrices in order to generate populations, which we can then use for things like differential expression analysis to figure out what genes are upregulated in those populations where certain clonotypes are present.
That would be good. And
Okay.
even the analysis, like you sent, it was a week or two ago, a nice little pinwheel diagram of VDJ. Actually, it was date, month, year, month, day, so forth. So you didn't use VDJ data. But is there a way to generate this pinwheel of what VDJs are?
No, we never did. We talked last time. We had considered including information on variable diversity and constant regions of the VDJ sequencing in one plot, but that's not present in the current iteration of the plugin. It does create pie charts that describe the differences there based on that statistic Jensen-Shannon divergence entropy.
Can you give an example of how to generate a pie chart? What would we do so you get?
Yeah, let's. It's not going to be interesting if we do just samples 165 and 166. So let's just run some really quick clustering. I'll use this Phenograph tool. Or actually, you know what? I think it's. Yeah, we could do this.
And do we have that file that he's talking about? Everything's together. Who has it? Do we have it?
This is just our.
No, but do we have the file that he's using? Everything put together. Because I don't have it.
It should have similar to that.
You're asking whether you guys have this analysis?
Even the file, yeah, the concatenated file. Just that
We have
No, I know we can make it, but he already made it. So it can, and Wei Chin saying he has it. I tried making it or looking around for it. I could not find it or make it. So it'd be, since you made it, could you make sure you send us that file?
Yeah, I thought I had sent you guys this combined file along
Yeah.
with the two pieces of annotation for VDJ, but I can resend that. That's no problem.
Right, that would be good.
Ian, just a quick question. So in regards, I know like the last few times, especially for 165, we were trying to just look at B cells. So,
Mhm.
like when we were trying to just focus the analysis on B cells, so like looking at the cell view where you can put, a transcript on the X and the Y, and how with the gating, how confident, will you? I mean, should you be, on setting the positive versus the negative,
Because of dropout events in
Yeah.
these single cell RNA-seq data sets,
Yeah.
we should be careful about calling anything using especially a Lin negative type. But what we could do is probably isolate, get a good guess at some B cells using like CD19.
Yeah.
We were doing that before. Did you say you wanted to focus specifically on sample 165?
Oh no. I was just saying. I mean, right? Like, so even if you have like a B cell population, and you said, okay, let's look at, what B cells express the transcript for CD27, which would be the marker for memory cells versus another, and then put on the x-axis like IGHD or something, and then trying, to sort of mimic the flow analysis,
Yep.
Would that hold up to statistical rigor?
Not sure what you mean exactly by statistical rigor. I think again, where you have like double negative cells in that example you gave,
Yeah.
you couldn't say for sure those are not memory cells.
Yes.
Right.
But for those that were CD27 positive, you might be able to say, these are probably at least a subset of the memory cells.
Okay. All right.
I
You know
can
how
show
to,
that.
but short of that, you kind of know how to do that.
Yeah.
Those two genes.
Yeah. No, it was just.
With that caveat that
Yeah.
negative you can't, might not be negative.
Yeah.
This is what we did before, right?
Yeah. I mean, yeah, and it's like, even like drawing a negative. I mean, I guess it's kind of clear here.
And even this one transcript, I'm not sure that cells that are expressing one transcript of CD19 are B cells. So I'm ruling them out. I'm throwing them into the double negative area.
Oh, the red is the
A
one.
little while to calculate. But basically, I'm going to say these are tentatively at least one population of B cells.
Yeah.
And then you mentioned you might want to look at within that a couple more markers.
CD27 and IGD or something.
IGHD.
Is that
Yeah.
right?
And then we could draw a similar kind of.
Yeah. I mean, like, I'm just thinking of, if we're going to try and publish that, and you said, oh, this was like the gating.
Well, it depends on what you want to do with that. I mean, I could identify, a gene set that I believe describes these 28 events.
Mm-hmm.
I could use that to go backwards and try and figure out, what else falls into that.
Yeah.
And it might turn out like we could utilize. I just ran UMAP here. Let's see if we got nice clusters there. I haven't even taken a look yet. Whoa.
Is that the cluster on the? Oh,
Oh, that's because I'm looking only at CD19.
Yeah.
But anyway, we could do some kind of clustering. Look at how those clusters differ based on the gene set for memory B cells,
Yeah.
for example. And then we also looked at IGHD, is that because you're looking for plasma cells, or what is that pointing to?
It was, you know, with like naive, it was just like a random example. You know.
Got it.
Naive are normally IgD positive, and memory may be IgD negative. And you know the graph sort of indicated that there's not many double positives, and there's a lot of IgD positive, meaning more naive cells. So it's sort of like representative, but you know we can't say what the negatives are. But you could say what the positives are if you, I guess, you know, we want to study that. I mean, you could get or a gene set because Wei Chin put together a gene set for something. I forget what it was. A cytokine. So gene sets are useful.
Yeah.
This or that, and call it, you know, basically.
Right. So, like, if I was going to do try and generate a gene set for the naive B cells, for example,
Right.
what I would probably want to compare with is anything that's not a B cell.
Yeah.
So again, make a not gate here.
Oh, not B cell or not naive?
It depends, but I was going to test against anything that's not B cell.
Okay.
Do you think it's better to take from the B cells anything that's not naive?
You know what I was thinking might be better is naive versus memory. And naive is whatever is naive but not memory, right? Because you know the T cell, well, there's a lot of them that aren't in the T cells. But if you do that by you do memory but not genes in memory but not naive, that might be a memory specific.
So if we're focused on memory, or sorry, naive, then I would put memory as the control on the x-axis,
Uh-huh.
and I would put naive on the y-axis.
Oh, right. Then you're back to this, the volcano plot.
Exactly. And these are going to be the genes that are up in naive
I see.
relative to the memory.
It's interesting. There's a lot on the other side.
Yeah, and actually the inverse is true as well in this case because we're just comparing these two populations. These genes over here are up in memory relative to naive.
Yeah, it's interesting. There's a lot. Can you look at those? I mean, make a gene set out of those? I guess you can, but I'm thinking that might be useful for us to do. Just to see. But I would have thought that a memory B cell, you know, sort of specialized in maybe has lower numbers of unique genes. But I'm surprised there are more. Yeah, naive has everything sort of. Maybe they're resting and they can't express it. I don't know. So those are the up in the memory. Wow, that's something. Or they make sense. See CD27 there.
Well, we defined them in CD27.
Exactly. So that
Oh,
should
right.
be-
So that's it. Okay. Right. I was just thinking of all the ones that were interesting. Like anything. Oh, CD44.
Yeah.
Yeah. Oh, CD40. Is this? Oh, it's alphabetical. Is there a way to show this in order of which or
Fold
more?
change.
Which one has the most?
Yeah, which one's more? I don't know. Is more significant? Kind of.
Yeah, there isn't. At some point, we'd like to, this is kind of looking forward, at some point, we're going to put columns in here, like value and fold change.
Right.
But currently, I think it actually does order these on fold change.
Oh, I see.
It's not alphabetical.
Oh, I see. Oh, okay. So CD44 is kind of the high pull. Is that kind of the more higher
And
pull?
their transcription factor, KLF6.
Which one?
KLF6.
I'm not familiar. But yeah, that's quite interesting.
Yeah. I mean, I guess, you know, I know you could do this. It's just, you know, with people that have, you know, more of a math background and, you know, with single cell analysis, you know, like with the caveat of the dropouts and